in Madison, where oddly enough he also grew up in that area. Talked about that earlier this morning. And then he joined the faculty in psychology at Wisconsin, and then he moved to NICHD in 1983. So my own experience with Steve is just generally this, that long before this interdisciplinary work became the fad that it is today, and the buzzword, Steve's been doing this for a lot longer. He's been using biological and social science data and theories emerging in very exciting ways. And I don't know how he's going to take it for this, but I usually introduce him and tell other people that he's the grandfather of gene by social environment interaction research. Partly because he like encapsulate lots of papers at the start of this area. You read those papers, especially the 5-HT2 volume paper, you'll recognize that almost all of it's based off of Steve's work. And so with that, on that big setup, I will turn the time over to Steve.
Thank you, Coulter. I'm not sure I'm ready to be a grandfather yet, but as I was explaining to Coulter, I have been getting over a very severe cold, and among other things, my ears are clogged, and so I'm having some balance issues. So if you don't mind, I'd like to give the talk while seated. Maybe I'll get excited in the middle and stand up and stuff, but in the meantime, I'd like to at least start. Can everybody hear me okay? All right. So, my laboratory at the NICHD, has been focused on study of individual differences in personality or temperament of the colony, within the colony of rhesus monkeys we're able to maintain at our facilities. Individual differences in personality and the biological basis underlying those individual differences in behavior and temperament. And in, as Coulter mentioned, in particular how genes and environmental factors act, and we think actually, much more importantly, actually interact to shape individual developmental trajectories. So as an example of what I mean by temperament or personality, we found over the years that about 20% of the rhesus monkeys in our colony, as well as 20% of the monkeys at 2 field sites we've had access to over the years, seem to be unusually fearful or anxious in the face of novel or mildly challenging stimuli. So situations and stimuli that most monkeys find really interesting and will readily explore, these monkeys tend to avoid. And if they are confronted with these circumstances, they show profound arousal of a variety of biological systems. The system we spent the most time studying is the hypothalamic pituitary adrenal axis that we index by looking at levels of cortisol, whether we're measuring it in blood or saliva or more recently in hair. These monkeys look very much like the group of children that first described by Jerome Kagan over 30 years ago, and subsequently studied by a number of people, Nathan Fox, my good friend at Maryland, being another, that Kagan called behaviorally inhibited. And we know that these behaviorally inhibited children are at certain risk, heightened risk for developing childhood anxiety and depressive disorders, and for some of them, this risk carries on to adolescence and adulthood. There's another 5 to 10% of the monkeys in our colony, and similarly 5 to 10% of the monkeys that we have studied in the wild, who seem to be unusually impulsive, and their impulsiveness gets them into trouble. They do stupid things that other monkeys are no better than to try, like get between a high-ranking female and her offspring, or they may repeatedly confront a dominant adult male, and when that male beats them back, they're right back in that male's face. And when that male beats them back again, they're right back in that male's face. So not surprisingly, they tend to elicit a lot of aggression from other members of their group. And they themselves show inappropriate patterns of aggression, in particular when they get into an aggressive encounter, whereas most monkeys know how to back off before things start getting out of hand. When these guys are involved, the intensity of the aggression tends to escalate to the point where somebody can actually get hurt.
And we have biological characteristics that differentiate these monkeys from others in their group. The characteristic we've spent the most time over the years looking at is an apparent deficit in serotonin metabolism that we index in terms of very low concentrations of the primary central serotonin metabolite, 5-hydroxyinositatic acid, that we see in cerebrospinal fluid. And as was the case with the uptight, as we call them, fearful monkeys, there's a human counterpart to these impulsive aggressive individuals, and these are kids who have been studied over the years by people like my colleague Richard Tremblay in Montreal and others, who find that there is a subgroup of children who already by age 2 are showing very high levels of aggression. And even though 2, according to Tremblay, is the period of the highest incidence of aggression in a human's lifetime, that's why they call them the terrible 2s, in most children the intensity and the circumstances under which this aggression is exhibited diminishes throughout development as they become socialized, except for this subgroup of individuals. And here, in them, the levels of aggression stay high throughout the early childhood years. They are still high when they hit the school system. They are often associated with classroom disruption. They're often diagnosed as having externalizing disorders. And by the time many of these kids get to their teenage years, you can find them either in prison or in the morgue. So we've been interested in these subgroups in particular, but other subgroups as well. First of all, because the features that differentiate them from others show up very early in life, and in the absence of major environmental change are remarkably stable from infancy through the childhood years, through adolescence, and even into adulthood. And we have increasing evidence that substantial components of these patterns are highly heritable. That is, clearly genes are involved. But we also have learned, and I'm here to try to convince you, that social environments are also important, and particularly the initial social bonds that individuals form with their biological mother, or in the cases of some research, some alternative surrogate or substitute mothers. And that the nature of these early attachments show up not only at the level of behavioral output and emotional regulation, but also at the level of neuroendocrine function, at the level of neurotransmitter metabolism, at the level of brain structure and function, and even at the level of gene expression and genome-wide patterns of methylation. So that's what I want to spend most of the day talking about. And I'll start by giving a little bit of background on my lab and the monkeys there. My lab is not located on the main Bethesda campus, main campus in Bethesda, but rather about 30 miles outside of Bethesda in the Maryland countryside near a little town called Poolesville. So when I mention Poolesville, the Poolesville colony, that's the one I'm referring to. And it's located on the grounds of the National Institute Animal Center, which is fairly close to the Potomac River. So here is the Potomac River. This is the Maryland side of the river. This is the Virginia side of the river. You may notice that there's a substantial amount of urban congestion, suburban sprawl. On the Virginia side, it's nothing but green on the Maryland side. That's what good zoning, strict zoning laws can do for you. And the animal center is outlined in yellow. It's about 1 mile square, 1 mile square on each side. And my lab is located in this red shaded area. So in contrast to the main campus, we measure our lab space in terms of acres instead of square feet. Within that facility, within our lab, we have a field station. This is an overhead view of it. It's a 5-acre enclosure within which a mid-sized troop of rhesus monkeys can free range year-round. And they are able to handle even winters like you guys have been having, and we've had in Washington, D.C. When it snows, the kids go out and play in the snow. So we have our monkeys living in a free-ranging, naturalistic setting where we can study them as if they were growing up in the wild. And when we do that, they are able to grow up in social groups that resemble what you'll find in the wild. And in the wild, rhesus monkeys live in large social groups ranging from 30 or 40 individuals on the small end to several hundred on the large end. But whether the groups are large or small, they all have the same basic social structure. Each troop is organized around several female-headed, multi-generational families or matrilines plus males who come in from the outside. And this arrangement derives from the fact that rhesus monkey females stay in the group in which they're born for their entire life, whereas rhesus monkeys stay only until around puberty, and then they leave home. They either leave voluntarily or they're physically kicked out of their social group. They then typically join all-male gangs for anywhere from several months to a year, and then eventually work their way into a new troop. So every troop has plenty of adult males and females in it, but the females have all been there since birth, and they're all related to one another, at least within their respective families. Whereas the males are initially related to no one until they start having offspring of their own. And here's one such family. Here's a mother, newborn offspring, grandmother's back there. This is the older sister of the infant. This is the younger sister of the mother. And this is characteristic as to whether the troops have many families or only 3 or 4. The other thing we should know about, and then this will come up later, is that within these troops there are multiple dominance hierarchies, differences in social status. So there's a difference between families such that all members of the highest ranking family, including infants, outranks everybody in the second highest ranking family, including adults, who outrank everybody in the third family, etc. So we see situations in the wild where infants, youngsters from high-ranking families who are barely able to walk may be stumbling down a trail, and adults from lower-ranking families will be falling all over themselves to get out of the way of these little kids, because they know if they cause these kids any grief, the rest of that high-ranking family is going to be on their back at all. So life is very different, even though these monkeys are in the same environment, depending on the social status of your mother. There are other hierarchies. There's a hierarchy within each family that follows the general rule: younger daughters outrank older daughters. There's a hierarchy among the males that come into a troop that's roughly related to tenure. That is, the longer a male is in a troop, the more likely that male is to be high-ranking. But it turns out that that's tenure and length of time is not what's really important. What's really important is how good those males are at making friends and sustaining alliances, not only with other males, but especially with high-ranking females who carry the real clout in the troop. And males that are good at that stick around and move up, and males that are not so good give up, leave, and seek their fortune elsewhere. And then finally, there's a hierarchy among the infants that are born into each group that roughly reflects the position in the troop of the mother. So it's not that some infants are bigger or stronger or smarter or quicker than other infants. It's that other monkeys associate a particular infant with a particular mother and afforded equivalent social status. So life in a rhesus monkey group is a pretty complicated business. And for an individual to survive in such a group, let alone thrive, it has to develop a fairly complicated social repertory, and it also has to learn a great deal about who else is in that troop, who's part of the troop and who's a stranger, where does each individual fit with respect to these multiple dominance hierarchies, as well as recent history, who's been getting into fights recently, who can you count on to back you up if you get into a fight yourself. And monkeys that are able to assimilate this information and follow the rules inherent in these multiple dominance hierarchies do very well in this complex society. Monkeys who are either unable to or unwilling to follow these rules don't last very long, at least not in the wild. So in a natural situation like this, rhesus monkey infants, who, by the way, grow up 4 times faster than we do, which is an enormous advantage for those of us who are interested in the long-term effects of early experience or lifespan development, or more recently, the transmission of characteristics from one generation to the next. We can see a generation in 3 to 5 years rather than having to wait 15 or 20 for the human equivalent. So when I give you an age in rhesus monkey months or years, just multiply by 4 and you'll get a rough human equivalent. At any rate, rhesus monkey infants spend virtually all of their first month of life in intimate social contact with their mother, and usually no more than arm's length away. And during this time, a strong and enduring attachment bond is established between mother and infant. It's functionally the same as the kind of attachment that, at least in Western societies, humans routinely form with their caregivers. Much of Bowlby's attachment theory was originally based on studies of rhesus monkey mother-infant interactions. So there's a quite strong similarity in at least the intensity of that initial interaction. And during that month, while these infants are developing this bond, the mothers are very protective about who they let interact with other monkeys. They usually don't let adult males too close to these monkeys, which is one reason in this species, not necessarily in other species, but in this particular species, males have very little to do with the caretaking of infants. They usually don't let unrelated females interact with their infants. And this particular photographer, mother, is not going to let the photographer get one step closer. Infants don't stay infants forever, however, and beginning in the second month of life, they start leaving their mothers for increasingly long periods of time. And they do this in a very stereotypic fashion, much as Bowlby described for human infants. They use their mother as a secure base. So they may wander away from their infants for a short period of time, and then go running back to their mother for a period of contact. Wander away a little bit more, and then run back to their mother for a period of contact. It's almost as if they're attached to their mother by a long rubber band. And by the time they are 6 months of age, these exploratory forays have increased to the point where they're actually spending only about 20% of their waking hours in physical contact with their mother, as opposed to 100% they were spending in that first month. Yet the mother's presence in the immediate environment is absolutely crucial to sustain this exploration, because as Bowlby has taught us, if you lose access to your secure base, any motivation to explore disappears and you become emotionally upset. So who are these youngsters interacting with during this time? Well, increasingly they are interacting with each other. And indeed, starting in the second or third months of age, these monkeys start spending several hours every day in active social play with their peers. And it's in the context of this play that virtually every behavior pattern that's going to be important for normal adult functioning can be practiced and perfected and become functional long before it has to actually become functional. So for us, social play is as important a part of the socialization process as that initial bond with the mother. And work by Harlow many, many years ago and by others more recently have demonstrated pretty convincingly that monkeys who are otherwise well socialized but denied the opportunity to play when they're growing up almost inevitably have problems with reproduction and almost inevitably have problems controlling their aggression. So for us, play is a pretty fundamental part of development. And then when these monkeys will spend much of the rest of their juvenile years, when they're 2 and 3 and 4 years of age, up to puberty, be developing and perfecting these complex patterns of play. And they basically learn how to interact with other monkeys in this context. Now, not all the monkeys at Brosville grow up this way. Some of them grow up with their biological mothers, but in smaller social groups. Here the infants remain with their mothers for their first 6 or 7 months of life, whereas other monkeys are run through a procedure designed by Harlow years ago that this gentleman was objecting to. They are separated from their biological mother at birth. They're hand-reared in a neonatal nursery with extensive human contact and stimulation, and then at about 1 month of age, if not a little bit earlier, they are put into small peer groups containing 4 or 5 individuals, and these peer groups, they stay in these peer groups 24 hours a day until they're about 6 or 7 months of age. At that point, we wean the mother-reared monkeys from their biological mothers, just as monkeys in the wild wean their kids at about 7 months of age, and we form large social groups containing both mother-reared and peer-reared monkeys from 7 months on until puberty. They stay in the same social group. So the difference between monkeys who we see in these groups, we can attribute to that initial 6 or 7 months of life when these different patterns of socialization were developing. And we now know that this has some consequences, behavioral and biological. For one thing, when these monkeys are growing up together, in that first 6 or 7 months of life, they spend an inordinate period of time clinging to one another rather than going out to explore their environment. So their exposure to their outside world is much more limited than they would have if they were being reared by their mother and going into this normal secure base phenomenon. Another consequence of these monkeys reared in this fashion is when it's time to start learning how to play, they do develop patterns of play, but these patterns never reach the level of sophistication that you'll see in their mother-reared counterparts. And when you think about it, it's not surprising, because these young monkeys have to serve as playmates and attachment objects at the same time, and they end up not doing a terrific job of either. And we find that when these monkeys get older and are in these other social groups, two features pop out. This is one of the social arrangements. This is an indoor-outdoor pen where there's groups of maybe, 20 to 25 individuals, basically a birth cohort for that part of the lab. And here's where they stay until puberty. And in these circumstances, we find that purebred monkeys seem to be unusual and anxious, and often show regressive infant-like behavior when they're put into novel or challenging circumstances. And in this respect, they look exactly like the 20% of monkeys in the wild who have this naturally occurring emergence of excessively fearful behavior, except they grow up in the wild, not in a peer group. And when these monkeys, just like those monkeys in the wild, we find they have greater output of cortisol for longer periods of time than their mother-reared counterparts under comparable environmental conditions. And we find so that they look very much, both behaviorally and biologically, like these fearful infants, the 20% of the population I mentioned before. But something else develops in these monkeys, and that is, as they get older, they start showing this explosive pattern of aggression in many of their interactions with other members of their group. Very much like the 5 to 10% of the population that I described at the very beginning of the talk, both in our colony and in the wild. And like that 5 to 10% of the population, they show deficits in their serotonin metabolism, whether you're looking at levels during the first year of life when they're fairly high for both mother and purebred monkeys, but they're still significantly different, as opposed to later in life when the levels have dropped for everybody, but if anything, the rearing condition difference is bigger. And although we know that individual differences within a rearing group are highly heritable, these differences are not due to genes, they're due to differences in their early social rearing. We've run these monkeys through, my colleagues in the Alcohol Institute have run these monkeys through the equivalent of a happy hour when they get to adolescence and young adulthood, and where for an hour a day, over a period of weeks and months, these monkeys have unlimited access to 9% NutraSweet flavored alcohol solution or non-alcoholic NutraSweet flavored beverage or plain tap water, so they're not fluid deprived. They consume this in familiar places, and under these circumstances we find some monkeys consume more alcohol than do others, and the ones that consume the most, as a group anyway, are those that spent their first 6 months of life growing up with each other rather than growing up in the more traditional family situation. More recently, we've been able to do neuroimaging of these monkeys, and we find that their brains are different. This is a PET scan. These are data looking images of monkeys going through a PET scan looking for serotonin binding potential on the left and cerebral blood flow. And what you have here is a comparison between a composite of a front view of a mother-reared versus peer-reared, composite top view of mother versus peer, composite side view of mother versus peer, and similarly for cerebral blood flow, mother versus peer, and mother versus peer. And simple inspection suggests that the peer-reared monkeys' brains are lighting up differently, are not lighting up as much as their mother-reared counterparts. And when you look at the actual data, that's exactly what you find. So, in sum, what you see in monkeys is a function of their how they were reared in the first 6 months of life. They show relatively normal behavior under familiar circumstances, but when they get under challenge, you have differences in their behavior, in their emotional regulation, in their neuroendocrine functioning, in their neurotransmitter functioning, and even in brain structure and function. And very recently, in a series of two collaborations with two groups, we started looking at gene expression and at genome-wide methylation patterns. So the first set of correlations, I should mention that we now have colleagues at the Yerkes Primate Center who have studied the groups of monkeys' mothers who, generation after generation, maltreat their infants, both neglecting and abusing them. It's about 10% of the population in the wild, and the people at Emory University, a group headed by Mar Sanchez and Paul Plotsky, have reported that you look at these monkeys who grow up with maltreating mothers and they look exactly like peer-reared monkeys in that they have higher levels of fear, higher levels of aggression, higher levels of cortisol, and lower levels of CSF5-HIAA. So in a sense, at least with respect to these behavioral and biological features, peer rearing is not a bad proxy for naturally occurring cases of neglect and abuse. And in the wild, this pattern spans generations. It's passed from one generation to another, although some brilliant work by Dario Mastapieri in Chicago has been able to demonstrate that this transmission is non-genetic, because when you do a cross-fostering study, the foster mothers, the kids who grow up, pick up the maternal style of their foster mothers, not their biological mothers. So almost certainly there's some kind of epigenetic pattern of transmission involved in this. Okay, so what about gene expression? My first collaboration, my lab is doing this with Steve Cole at UCLA as a molecular biologist, and with James Heckman, the Nobel Prize winning economist from the University of Chicago. And what Steve does is heat plots. So he's looking at samples obtained of white blood cells, lymphocytes, and looking for patterns of gene expression. And he comes up with data that look like these. We have published these data. Let me unpack it a little bit. What you have in each bar is a different gene. What you have in each column is a different gene. Each line or row is a different monkey. So this is mother weird 31, mother weird 45, mother weird 43, mother weird 41, mother weird 55, and then period. If you see red, that gene is overexpressed. If you see green, that gene is underexpressed. And if you look at that, there's almost no overlap in these two groups of monkeys, even though, and these data were obtained when the monkeys were 4 months of age. With one exception, there is no overlap except for MR55, who, although being mother weird, actually looks more like a pure weird monkey. And it turns out that MR55's mother was a maltreating mother, and again showing the same pattern of gene expression that we see as a function of differential early rearing. Now, this was a one-shot sampling when these monkeys were 4 months of age, but we have since sampled other monkeys longitudinally from birth up until puberty, and those data are just about ready for prime time. I haven't gone over the details of it, but I can assure you that when we make major shifts in environment, such as moving them from their original rearing group into new groups, we get dramatic changes in the pattern of gene expression. These are epigenetically generated things that you can attribute to particular environmental manipulations. Now, what are these genes doing? I can't tell you, but Steve Cole can. And he finds that many of these genes that are underexpressed in pure weird monkeys have to do with immunoglobulin production, type 1 interferon, antiviral responses, antibacterial responses, whereas the genes that are overexpressed in these pure weird monkeys have to do with inflammation and cell growth and differentiation and transcription control. These are just some of them. So that, you can say, well, that's nice, but what does it mean? Well, Steve Cole's day job is not looking at monkey mRNA, it's looking at human mRNA, that in collaboration with John Cacioppo at the University of Chicago. He's done similar studies looking at adult humans who characterize themselves as feeling lonely or socially well integrated. And you get a heat plant that looks remarkably like the ones that you see differentiate mother weird and pure weird monkeys in terms of the ones that are overexpressed in the pure weird monkeys are exactly the same as the ones who are overexpressed in the mother weird monkeys, and similarly the ones that are underexpressed in the individuals who consider themselves to be socially to be lonely are the same genes that are underexpressed in the pure weird monkeys. Now, I'm not about to sit up here and tell you that pure weird monkeys are just like people who characterize themselves as being socially isolated and lonely, but I think you have to admit that the correspondence is really quite striking. The other type of work that we're doing is in collaboration with Moshe Ziff, and this is looking at genome-wide patterns of methylation. In our initial study, we looked at both a different type of white blood cell. It was leukocytes for Steve Cole, it's lymphocytes for this collaboration with Moshe Ziff and his colleagues at McGill University, and we were able in the initial study to get samples from adult monkeys who had been differentially reared for the first 6 months of life, and then had been in the exact same physical and social environments until they were 8 years of age, when they were put into a terminal study by another group of investigators. This is just showing what happens when a cell when a gene gets methylated. And here's Moshe Ziff. Here's a younger version of Moshe Ziff. He's lost a little bit he's become balder since, I can say that. And he looks at genome-wide patterns of methylation. And he comes up with things like this. So here's chromosome by chromosome. Here's prefrontal cortex, tissue from prefrontal cortex. Here's white blood cells, lymphocytes. If the bar is above, each bar is a gene. The bar is above the line, it's more highly methylated in mother reared. If it's below the line, it's more highly methylated in peer reared. So this is all the genes differentially methylated on chromosome 1, chromosome 2, etc. I could show you each of the chromosomes, and you'd see that across the entire genome, fully 4 to 5,000 genes are differentially methylated. That's 1/4 to 1/5 of the entire rhesus monkey genome, just as a function of what happened in that first 6 or 7 months of life. Now I can go, here's the chromosome X, you see the same pattern. And one of the things that Moshe has found in his analyses is that these genes, the experience effect on these genes tends to be clustered both in terms of function and in terms of position on the actual chromosomes. So there is some correspondence between genes that are methylated in blood and genes that are methylated in brain. It's far from complete. It's quite tissue specific, but maybe 30% of the genes that are differentially methylated as a function of that early experience are the same genes in both brain and in blood, then in the same direction than you see throughout the rest of the genome. So what this means is that while there is clearly tissue specificity, and there's a lot of it, there are a few genes for which we can get some idea of what's going on in the brain by looking at peripheral measures such as can be found in blood. And the reason that's important is that you can do the brain measures only once, but at least with current technology, but you can take blood every week. And that's in fact what we've been doing, and we have almost as ready for prime time the same notion of what happens when you look at these differences that are so prominent early on throughout later aspects of development. The other thing is that these genes tend to cluster by function. So these are genes that are differentially methylated as a function of being in the serotonergic pathway, and this is in T cells and in prefrontal cortex. And you can see that for some of the usual subsets, serotonin transporter, serotonin 2,8 receptors, you have effects that are same in blood and brain, whereas in MAOA or MAOB, you get different effects depending on what tissue you're looking at. This is again at 8 years of age after these monkeys have been living under the same circumstances for 7.5 years. You see an even more complete pattern when you look at genes that are differentially methylated between mother-reared and nursery-reared monkeys that are part of the HPA axis pathway. And once again, we have some specificity. So all the vasopressin genes, you only get an effect in prefrontal cortex, but for many of the cytokines, you can find similar effects in both particular areas. And so now as we learn more about what these genes are actually doing, we can start talking about pathways and what these methylation patterns and expression patterns are really signifying. Finally, we have since begun some intervention studies where we can go in and try to see if we can reverse some of those early experience effects. And we've used a number of different techniques, but the one that we know the most about involves changing the environment, simplifying the environment for these monkeys when they move from their original rearing group into these large groups of mixed, where you have both mother reared and pure reared being in the same situation. And one of the things we've done is made the group sizes much smaller. We've introduced them a little bit earlier, so at 6 or 7 months instead of at 8 or 9 as we originally do. And we put into each of these groups what we call foster grandparents. These are very old females, post-reproductive age females, who, when they were growing up, were exceedingly nurturant of their own offspring, and old males who basically are there to keep the peace. They will break up fights that any of these youngsters might get into. So here's one of our old males. This is Doogie, and I can assure you nobody's going to mess with Doogie. So in this environment, we find that some of these early differential rearing effects appear to be getting reversed. And I'll just show you the first set of the longitudinal data we had. This is looking at simply the number of genes that are differentially methylated as a function of whether they grew up in the nursery or whether they grew up with their biological mother, and broken down by males and females. And you see that already by 1 month of age, there are almost 5,000 genes that are differentially methylated in both mothers and infants. It makes a difference in terms of gene methylation, whether you're being cared for by human caretakers or whether you're being cared for by your biological mother. And it's about the same for males and females. But look at what's happened by the time these monkeys are 2 years of age. The intervention happened right here in the middle. And unfortunately, we don't have 1 year samples. We had 1 year samples, but we unfortunately shipped them to McGill over the Christmas holidays, and they got stuck in customs, and they all melted. So it was like getting a lump of coal in your Christmas sock. Very disappointing. But even if you look at the 2 year data, you can see that in males by 2 years of age, it's not 5,000 genes anymore. It's 2,500. It's half of what it used to be. So what has happened is many of the genes that are differentially methylated now have the same pattern, overcoming that early rearing effect. And look at the females. For females, it's not 5,000, it's not even 2,500, it's 750. Now, 750 is still a significant number of genes, but it is a fraction of what it was in much earlier in life. So something is helping to reverse this, and we think what's going on with the females is this is the end of 2 years is you see the very first stirring of puberty appearing. And it may be that with the onset of puberty, you get a recalibration of these things, and it may be that some of these genes coming online are now more important